Today we're going to be talking about translation, or protein synthesis. And translation is the process by which we take information in the messenger RNA and convert that into a protein. And this occurs in the cytoplasm of the cell. So this is our messenger RNA right here. In the previous video we made that messenger RNA. It was made in the nucleus, it left the nucleus through the nuclear pore, and it's now in the cytoplasm. Once this messenger RNA gets into the cytoplasm, that's where translation is actually going to occur. And a ribosome is going to recognize this messenger RNA. And it's going to come and it's going to attach to the messenger RNA. And that's going to start the process of translation. But let's just go over some of the parts here. So we've got the messenger RNA. And the messenger RNA is actually going to be made up of sequences of bases. These sequences of bases, and the messenger RNA is actually going to be read 3 bases at a time. And each sequence of 3 bases is referred to as a codon. The ribosome is illustrated here in green, and we labeled that ribosome. This is a large protein containing ribosomal RNA. In fact, the ribosome is actually about 2/3 ribosomal RNA and 1/3 protein. This ribosome consists of 3 grooves, 1, 2, and 3. The first groove here is referred to as the A site. And the A site is where the next incoming transfer RNA with its attached amino acid is going to come in. The P site represents the protein site. This is where the growing protein is going to be made. And then we have the E site here. E stands for exit. So once the transfer RNA molecule gives up its amino acid to the growing protein, that empty transfer RNA will then exit this E site, go out into the cytoplasm, and attach to another amino acid. In blue here I have the transfer RNA illustrated. And the transfer RNA has a sequence of 3 bases here called an anticodon. And this anticodon, this sequence of 3 bases, is actually complementary to the sequence of 3 bases on the codon. And that's how they recognize each other. They actually pair up through hydrogen bonding, which is a weak type of bond. So let's go ahead and begin the process of translation. So our messenger RNA has found its way into the cytoplasm. A ribosome has recognized this. The ribosome has gone over. It's actually attached to the messenger RNA, and it's looking for a sequence of 3 bases called a start codon, which is our very first codon. And there's only one start codon, and it consists of a sequence AUG. So the ribosome starts looking for this sequence of bases AUG. And here it is right here. Once the ribosome finds this AUG, this start codon, this is where it actually sets up. This is where it assembles itself, and the other parts of the process then come in and join the ribosome. So here's AUG. That's going to be our start codon. Our first transfer RNA with attached amino acid comes in, and it pairs up with AUG via hydrogen bonding. Now U is going to pair with A is going to pair with U, and C pairs with G. So this is the only transfer RNA that can pair up in this particular place. And attached to this transfer RNA is the amino acid MET, which stands for methionine. It's one of the 20 amino acids. And we labeled that AA amino acid. So that's going to be our first amino acid. And how do we know that MET should be attached to this transfer RNA? Well, it turns out that scientists have actually worked this out. And there is an actual chart called the genetic code that shows us how to figure out what amino acids should be in what place. And these amino acids go according to the codon. So this codon is AUG. So I'm going to look at my genetic code table here. And right here, the left side is the first base in the codon, the top is the second base in the codon, and the right side is the third base in the codon. So my first base is an A, so I'm going to go to the left side and I'm going to look for an A. Here it is. The next base is a U, so I'm going to go to the second base and look for a U. There it is. And I go down and over and I come to this box. There's 4 amino acids in this box. ILE, 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 MET. So how do I know which one of the 4 it is? Well, I'm not done yet. So I'm in this particular box here. So I've got to go over to the right and I've got to look at the 3rd base in the codon, which is, in this case, G. So I go, here's the box that I am in so far. I go over to the right and I got UCAG. I'm going to pick the G. And I follow that G over to the box that I'm in, and I see that it lines up with MET, which stands for methionine. So MET is going to be our first amino acid. The next thing that's going to happen in this process is another transfer RNA with its attached amino acid is going to come in to the A site. And once this happens, we're going to attach our amino acids together. But again, how do I know which amino acid would be attached to the transfer RNA? Well, again, let's take a look at the codon here, the second codon, AAG. So I'm going to go up. Here's an A. There's the second base over here, A. I go down and over, and I end up in this box, ASN, ASN, LYS, LYS. Those are my 4 amino acids to choose from. So I'm going to go over to the right, and I'm going to look at the next base, which is a G. And the G, if I bring it straight over to the box I'm in, lines up with LYS. So the next transfer RNA that comes into the A site would carry the amino acid LYS, which stands for lysine. And this process would go on and on and on. Now after this next amino acid comes in here, we're going to attach these 2 amino acids together. And that occurs via an enzyme. Once those 2 amino acids are attached together, then the ribosome moves forward 3 bases, and the next transfer RNA with its attached amino acid comes in here. At the same time this is happening, that empty transfer RNA releases and goes out into the cytoplasm and gets another amino acid to attach to itself. Now this is going to occur on and on and on and on until we get to another special sequence of bases. And this special sequence of bases is called a stop codon. And it turns out there's 3 stop codons, one of which is called UGA. So if we go on and on and on, we eventually come to here, a stop codon, UGA. Now there is no transfer RNA that will pair up with UGA. So when that happens, the ribosome recognizes this and releases the protein, it releases the ribosome, and everything goes their separate way. And our protein is almost finished. Eventually at that point it's going to go on for what we call post-translational modification. That's some modification that goes on after the protein is built. So how many bases did it actually take to make this particular protein? Well, it took 3, 6, 9, 12, 15, 18 bases to make this particular protein. Now the protein was only made from the information from 15 of these 18. But we have to include the other 3 simply because they're needed to tell the ribosome to stop the translation process. So in this case, we have 1 amino acid, 2, 3, 4, 5 amino acids. So our protein is actually 5 amino acids long here. Now in real life, most proteins in the human body are around 500 to 1,000 amino acids long. Some are shorter, some are much, much longer, upwards of even 1 million amino acids long. And that is it for protein synthesis.